Welcome to part 2 of the membrane structure and transport video. In part 1 we looked at the structure of the membrane and those first two modes of transport, and this video we're going to look at the other three types of transport across the membrane, starting of course with osmosis, as we see in front of us here. Really important that you know this definition of osmosis. It's one of the few definitions that you just have to rote learn. Just remember this one, that's all I can say. It is defined as the net movement of water molecules. So osmosis is purely about water, so you've got to say the movement of water molecules in there across a partially permeable membrane. So most of the time we're thinking about the cell membrane there, from a region of higher concentration to lower concentration. But we've got to stipulate that that is of the water molecules, because whenever you're dealing with osmosis, you're talking about water, but you're also thinking about the solute concentration as well, the stuff that is dissolved in the water. So if water has a high concentration at one point, a relatively high concentration, then the solute will have a relatively low concentration, because it's all a relative balance between the water and the solute. So if you just say higher to lower concentration, it's not clear whether you're talking about the solute or the water, and really they're going to be opposite to one another. So you have to say of water molecules. So yeah, just learn that definition. So you can see that solute concentration is a super important thing when it comes to understanding osmosis. So important that we have a special name for solute concentration. It is tonicity. So down here, tonicity is the concentration of solutes in a substance. Understanding tonicity is the key to understanding osmosis and predicting the movement of substances in or out of the cell. So let's look at different tonicities. If you're in an isotonic environment, it's an environment where the extracellular fluid, that is the stuff outside of the cell, has the exact same solute concentration as the cell, as the cytosol of the cell. So that's isotonic, the same concentration on either side. In that case, there'll be no net movement of water in or out of the cell. Water will still move in or out of the cell, but it will happen at an equal rate. So there'll be no overall or net movement of water across the plasma membrane. Therefore, the cell will keep its given structure. Let's look at animal cells and plant cells separately, because the outcomes are slightly different as a result of osmosis. If the animal cell gets put in a hypotonic environment, and that is where the solute concentration of the extracellular fluid is lower than that of the cell's cytosol, then that environment is hypotonic, lower concentration on the outside than inside the cell. In that case, water will move into the cell. Water will move from areas of low concentration of solute to areas of high concentration of solute. You can see how getting that solute versus water concentration thing is important. The other way of thinking about it is water moves from a hypotonic environment to a hypertonic environment. Because if the surrounding fluid is hypotonic, relative to that, inside the cell will be hypertonic. It will have a relatively higher concentration of solutes inside than there is outside. And so water will move into the cell in that case. If it's an animal cell, then that can be a problem if it doesn't stop, because if water keeps moving in and there's nothing to stop it, the cell may burst. It may undergo cell lysis. That can be quite a problem. If we look at the exact opposite situation, if the cell is put into a hypertonic environment, this is the case where the solute concentration of the surrounding fluid, the extracellular environment, is higher than that of the cytosol, the interior of the cell. So in this case, because it's a relative measure, the outside of the cell is hypertonic, high concentration of solutes. The inside of the cell will be hypotonic, relatively low concentration. Therefore, if we follow that principle of water moving from a hypotonic to a hypertonic environment, water in this case will leave the cell. It will move out of the cell, and the cell will shrink in as a result, because a lot of the bulk of the cell, of that cytosol, is water. The cell will shrivel and will undergo a process called crenation. The principles are the same for plant cells. This hypotonic to hypertonic movement of water will hold true as well. So if a plant cell is in a hypotonic environment, again, water will move into the cell because the cytosol will be relatively hypertonic. But a plant cell won't burst. It won't undergo cell lysis because it's surrounded by a cell wall, and that cell wall will keep it will keep that cell intact. The cell in that case will be termed turgid, meaning that the cytosol and the membrane are pushing out against the cell wall, causing it to flex, but it won't cause it to burst. It's really important for plant cells to have this cell wall because they often find themselves in watery environment, and they don't want to constantly, if they were to constantly take that water up, then they wouldn't want their cells to burst. They can't run away from the watery environment. Sometimes they'll just be stuck in it, and so they need an adaptation to help them, not burst in that situation. And again, hypertonic plant cells are the same as animal cells. In this case, the surrounding solution is hypertonic, so it has a high concentration of solute. The inside of the cell, it will therefore be hypotonic. So water will move from the hypo-interior to the hyper-exterior. It will leave the cell. However, the plant cell here won't shrink down in overall appearance because the rigid cell wall will allow it to still keep its structure. The state here is known as plasmolysis, or the cell has become flaccid. Again, this is an important aspect for plants. They don't want to completely shrivel down because they rely on their ability to stand up, I guess the plant does, and still capture sunlight even though they're in a low water environment. If the plant cells were to completely collapse and shrivel down, the plant would just collapse in on itself and it wouldn't be able to collect sunlight, and that would be a bad thing. It would then die before it can get another drink of water. Okay, so that's osmosis, our last form of passive transport. Then move into the active forms of transport. And there are two, active transport and cytosis. But active transport that term is often used to describe the movement of substances across the membrane through protein channels, specifically against their concentration gradient. So active transport is performed by specific carrier proteins. These are those transmembrane proteins. It's similar to facilitated diffusion in that respect, except it's against the concentration gradient. Once again, the shape of the protein channels is specific to particular molecules, so the cell can control what it will actively move in and out of the cell by having those specific protein channels present. The cell will use ATP to power these protein pumps, sometimes referred to, that allow it to move molecules from areas of low concentration to areas of high concentration. As I said, these are sometimes referred to as membrane pumps. They are because a pump works to use energy to transport something from an area where there's where there's lots, but I guess the idea, the analogy of the pump is that it uses energy to transport something, I guess. And we'll come across these more when we look at how a nerve signal is transmitted. This one here is a sodium-potassium pump, and that's really important in a nerve cell. And this looks like it's a proton pump, which is not as important to us, but we'll see them every now and then. Okay, the last form of transport is bulk transport or cytosis, and there are two main branches that we'd look at here. First of all is endocytosis, describing the movement of substances into the cell. Endo meaning into. I'm sure there's other words that use that endo prefix, but I can't think of them right now. But anyway, there's substances on the exterior environment. The cell really wants those. Maybe it's some food, maybe it's a pathogen, a bacteria that the cell wants to kill because that's its job. It will use endocytosis to take that thing inside. It requires an input of energy because it's a pretty involved process. And we can branch it down into two different types. Phagocytosis, engulfment of solid particles. We'll particularly be looking at this more in immunology when we look at specialized phagocytotic cells. And pinocytosis, we won't be looking at anymore. It's just the same thing, but with liquid, with a fluid. Here's a nice little flow diagram of that process happening. There's some solid particles hitting the outside of the cell here. And the cell will have receptors on the outside that allow it to recognize that that's something that it wants, to, to take in, and therefore we'll start to see this folding in of the membrane. And move over to step 2, the next one to the right, it's folded in almost all the way around, and then it's pinched off this vesicle from the membrane. Again, this is key because the membrane of the vesicle, membrane of the cell are made up of the same thing, phospholipid bilayer, so it can easily bud that off. It might then go and merge this vesicle with a lysosome, and that's not a problem because a lysosome is also a membrane-bound vesicle, so they can fuse together, and the enzymes, the digestive enzymes in the lysosome, could then break down those particles into small things that the cell might then recycle, for example. Exocytosis is the opposite. This one's a bit clearer, exo being the exit of things from the cell. So that one makes sense. At least if you can remember that, then you'll understand that endo it means to take in. So this is about releasing substances out of the cell, and it's really just the exact opposite of that process of endocytosis. If you look at the flow diagram here, I think whoever made this diagram probably just saved time and flipped them, just reversed it, because it's the same thing. We've got a vesicle, and this vesicle might have come from the Golgi apparatus. It might be containing some useful digestive enzymes that we want to use in the digestive system. It might be containing some useful hormones that we want to send as a signal to somewhere else in the body. They're packaged in a transport vesicle. That vesicle fuses with the membrane because they're made out of the same stuff, and the contents then get released into the environment to travel off into the bloodstream or wherever they may go. Okay, and that is the last slide, which means we've come to the end of membranes and transport. There are a few things that we can pick up on in more detail in class, but those are the key points that you need to be across. So thank you very much for your time. Again, feedback always appreciated if you feel so inclined. Okay.